Okay, so hello everybody, and welcome to today's talk in our Evolution and Ecology seminar series. My name is Emily Fowler. I'm a postdoc at the University of East Anglia in the UK, and I'm part of the new organising team for this seminar series. My co-organisers are Sarah Worsley, also from the University of East Anglia, and Valentina Piona and Aaron Fogan from Uppsala University in Sweden. On behalf of the new team, I would like to take this opportunity to say a huge thank you to the original organisers, Elizabeth, Wouter, Julia and Andreas, for all their hard work in putting together such a fantastic series over the past year. As we transition to a new team, we have one more talk scheduled on the 28th of July, where we will be joined by Dr. Yasir Ahmed-Brahmar. We then plan to take a summer break over August and return in the autumn with a new schedule of talks. In the meantime, we have an archive of recorded presentations on our YouTube channel, so please feel free to catch up on any talks you may have missed over the past week. So now on to today's talk. As always, there will be a Q&A session after the talk, so please do post your questions in our designated Slack channel. If you haven't already joined, you can find the link to our Slack channel in the description below this video. Today, we are delighted to have with us Dr. Tristan Long. Tristan is an associate professor at the Wilfrid Laurier University in Ontario, Canada. Tristan obtained his undergraduate degree from the University of Western Ontario, his master's from the University of Guelph, and his PhD from Queen's University in Canada. After his PhD, Tristan held postdoctoral positions at the University of California, Santa Barbara, and the University of Toronto before taking up his current position at Wilfrid Laurier. Tristan's research covers a fascinating array of topics in the fields of evolutionary genetics and behavioural ecology. Using experimental evolution, behavioural assays and genetic techniques, Tristan has conducted high-impact work on sexual selection and its implications for reproductive success, and the genetic and environmental sources underlying phenotypic variation. Tristan's recent work includes studies on the role of relatedness in mediating conflict and cooperation, plasticity in female mate choice, and the effect of the social environment on oviposition and foraging behaviours. Today, Tristan will talk to us about conflict, cooperation, and constraint in the fruit fly Drosophila melanogaster. So Tristan, thank you very much for accepting our invitation to speak today, and it's over to you.
Well, thank you to you and the rest of the organizing committee. It's a great honor to be presenting you today. So the work I'm going to be talking about was work that was conducted here at Wilfrid Laurier. For those of you that are familiar where that's located, southern Ontario. And Laurier and its campus are located on the Haldeman Tract, the traditional territory of the Neutral, Anishinaabe, and Haudenosaunee peoples. The work that I'm going to be talking about today was funded with a discovery grants from INSERC, and would not be done if it wasn't for all the hard-working students that have been part of my lab over these sort of last 10 years. Today's talk is going to highlight some work done by 4 of these students, Heather, Lucas, Tara, and Matt. Of course, the work that we're going to talk about today would not also be possible if it wasn't for my writing as well. Now, as Emily mentioned, the work that we do here in the lab sort of straddles the border between ecology, behavior, and genetics. And my particular interests are in things related to sort of sexual selection and sort of sexual conflict. And the model in which that I have been doing most of my work for these last 20 odd years has been working on sort of fruit flies, which are an absolutely amazing model system, great history, but also just on their own sort of qualities are really exciting because they show much complex behavior. They have really interesting sort of social dynamics, and so even after 20 years since Adam Chipnell first gave me my first sort of bio of flies, I'm still excited about learning more about them and using them to sort of understand the underlying sources of variation in the behavior, and understanding sort of the consequences and the evolution that can tell us of larger processes. Now, well, my particular interests are in sort of the causes and consequences of variation in female choice and sexual conflict. I do encourage my students to try new things and push the envelope out of my comfort zone. This is something that I was very privileged and lucky to have the opportunity to do so early on in my research career. And so, while my sort of core interests are in those sort of topics, that often my students will go and try different things to expand our knowledge of them and sort of discover fascinating stuff. So today's talk is going to focus on sort of three different projects. We're going to talk about Heather Mallett's work, looking at social cues used when selecting overpositioning sites. We're going to talk about Lucas Kodehi's work on cooperative foraging in larvae, and we're going to round it out by talking about some experimental evolution work done by Matt Maloney and Tara Newman that is looking at the potential constraints on the evolution of sexual values. Now, we'll begin with Heather's work, and Heather was really interested in understanding sort of the social dynamics of decision, because when one makes decisions, one has to bring in lots of information. One might have to store it, might have to judge it, and that can often be a huge investment of time and energy. For instance, in this cartoon, if we imagine that we have this female here that is assessing different males, the information that she acquires via her own experiences, trial and error, are her private information. However, in many species, including Drosophila, there's evidence that individuals are using social information, public information, looking at decisions made by others and using that to determine the decision. And there are, this is a fairly widespread phenomenon. It's thought to be an adaptive phenomenon, possibly because it requires less investment of time, energy, and resources. It may allow you to make faster decisions, but of course it does come with some risks. The risk, of course, being that copying other individuals might be deleterious if that information you're acquiring is of low quality or even sort of irrelevant. So, while Heather initially was interested in mate choice copying, which is a burgeoning field in Drosophila. There's actually an interesting paper that just came out this week in Animal Behavior on the subject. She was in fact decided to look at egg laying or overpositioning decisions, because that is an important decision that an individual makes that have huge implications for the success of the offspring. And she was strongly influenced by the work of Golden and Dukas, who did a really interesting study in which they placed overpositioning female fruit flies into a chamber. And at the bottom of the chamber, there were two small petri dishes that contained food. In every chamber, there was a reference food, which contained standard 100% nutrition food, and had not had any contact previously with fruit flies. The test food, which was the other petri dish in the chamber, was manipulated in two different ways. There were three different nutritional treatments. The food either had 100% nutrition, 30% nutrition, or 11% nutrition, and it had either been exposed previously to overpositioning females. So females had been placed onto these dishes and allowed to lay eggs and associate with the surface of them, or the petri dish had not had any exposure at all. So that's the social versus non-social treatments. And what we sort of they saw was that in the 100%, when the test food was 100% and had no social contact, it was used as much. It was used as much as the reference food. So right there, sort of that 50%. However, that same food, if it was of lower concentration, was used increasingly. So this again is females using private information and avoiding laying eggs on when given a choice on low quality. Now, what Golden and Dukas found was really fascinating was that when the test food had some previous exposure to ovopositioning female, almost over 80% of the eggs that were laid in the chamber were laid on that food, even though it was of the same nutritional quality as the reference food. So this really suggests that sort of public information, that social information, was being used extensively in making decisions, and was in the other treatments they found that that even the social food that was 33% nutrition got almost as many eggs as the reference food, which was 3 times more nutritious. So this really suggests that fruit flies are using this social information that's derived from conspecifics, others in their environment, and that overrides their own sort of personal information. So this really sort of captured Heather's interest, and so she started sort of brainstorming with different sort of questions that she could ask based on this. And one of the questions that she was particularly interested in was sort of the how well these females could identify relevant social cues. Golden and Dukas suggested and found evidence that these cues may be affected strongly by the microbiome of the demonstrator females that oviposited and added these scents onto the food. And so she set out to sort of see, well, what happens when we have females that have a choice of many different kinds of smells. And the first kind of microbiota cue scent experiment we did used our focal flies, Drosophila melanogaster, from the Ives population. And in our sort of readings of microbiomes, we saw that not only are there sort of differences between the microbiomes and thus potentially the cues that are associated with different species, and in our lab we have populations of Drosophila suzuki and Drosophila simulans that we sort of pulled into the experiment. But we could also sort of saw that diet often influenced the microbial community that the fly carries in the digestive tract. And so we raised fruit flies on both our standard fruit fly food, which is a nice banana medium, as well as on one which there was a high protein, low carbohydrate diet. So thus, with these sort of different combinations, we have sort of five different treatments that demonstrated treatments. Starting from the bottom, we have our control chambers, where we just had a chamber with standard food, which we didn't add anything in. In the next treatment, we added in the Drosophila suzuki, males and females, to oviposit. We had brown-eyed Drosophila similans, which we added in our third treatment. Our fourth treatment used standard IV females that were mated to males that carried the brown dominant marker, and we did that so that we could distinguish between our sort of focal females, offspring from any of these sort of potential other eggs that might be laid on the surface. And our final sort of treatment were these IV24 females. So these are females that were raised on this high protein feed, and we also mated them to the same type of IV BWD or brown dominant. So we give them a few hours for them to pass on their scents and associate with these discs of food. We then move them into a cafetiere style chamber, and then we placed into those 10 target females. In this first assay, they were also IV24 females that were already mated. And then we monitored them for 5 hours, every half hour, and went through to sort of see how many individual females were on each one of these 5 different blinded chambers, and we summed them after 5 hours. At that point, we then took them all out, placed them into a new vial, and let them develop for 14 days, and counted the number of offspring. The IV24 females all will have red eyes, well, it's easy to distinguish all the other eggs that might have been laid on those individual chambers. Here's a picture of that in action of some of the students who are working our way through this experiment through a long day. And here's a picture of one of our cafetiere style chambers, so you get an idea of what is happening. And what we found initially was that, consistent with sort of with Golden and Ducas, that although all the food in all these different discs was the same, that females associated far more with those that had been exposed to any kind of Drosophila. That they avoided the controls, or more accurately, they associated more with the other sort of 4 different petri dishes that were in the chamber. But what was really fascinating was when we took all those discs, we grew them up, we waited 2 weeks, so we found that almost all the eggs that were laid were on that IV24 exposed dish. So females were pretty much just spending all their time laying eggs on that one, which is the same, which had been exposed to females, which had the most similar history to our IV24 focus. Now, when we saw this, we thought, okay, well, maybe there's something weird about IV24 females. Maybe in their preferences for things, maybe in terms of the particular cues of growing up on that high protein diet. So we did the entire experiment again, with just regular IV females, and with the same kind of exposures. Now, and we sort of saw again when we just looked at the observation at the observations that we saw, similar as before, that those that had been exposed to any kind of Drosophila got a little bit more attention than did the control populations. But what was fascinating was that when we looked at the number of eggs, and we let them lay overnight this time just so we could have a greater chance of getting more offspring, we saw that the most eggs were laid on the IV BWD disc, which were IV females that had been mated to IV BWD or brown-eyed. So these were, in this particular case, these were the individuals that were most similar to the focal female. The IV24s have been developed in the regular food, and the IV BWD females have been raised on food. So that they did not show that strong preference for the IV24s. So this does suggest to us that females are very sensitive to the microbial cues, the sense, and the signals that are being left behind by humans. And this information is a really important driving force in determining where they're going to lay their eggs, which is a really important decision. Now, from here, we decided to sort of to ask more bigger questions about is the information that is important or relevant for making a decision for a female fruit fly, if she is going to use information made by conspecifics. Well, because not every individual might provide the same quality or relevant information. So if we're expecting this to be true copying behavior, where individuals would be using the most relevant information for deciding what is a suitable oviposition site. Well, then we made some I think, reasonable predictions that given the choice between female demonstrators and male demonstrators, that female demonstrators are probably more likely to provide relevant cues as to the suitability of a site for oviposition than males. That was one of our sort of predictions, assumptions. And building on that, you'd expect that those females that had mated would provide more useful information than unmated individuals. And so you might expect that, all else being equal, that you would expect more females associating with demonstrator areas that were female over male, and mated females over virgin females. And we also sort of realized that the interest in using these sites may also differ based on the focal female's state itself. Virgin females and mated females differ in terms of many different things, including nutritional preferences. And so we also were interested to see whether if we manipulated the mating status of the focal flies, whether they associated differently with the different options in a sort of cafeteria style experiment. So in this particular experiment, we then had 5 different treatments where we had control chamber with no exposure to females. We had virgin males that had been exposed, mated males, virgin females, and we also for our 5th treatment we used regular IV females that were mated to our spermless male, so they couldn't produce any viable eggs. So flies could not use sort of the presence of any larvae as a cue. So after giving the sort of petri dishes some exposure to these types of flies, we then sort of placed them into these arenas, and we either placed in females that were either IV females or mated IV males or virgin females. We observed these chambers for 6 hours and counted the cumulative number of females that were seen on each one of these 5 different kinds of dishes, and let them lay overnight. We then removed those dishes and let them grow and counted the number of offspring. Now, we saw this is one of those experiments where all our well thought out predictions were all sort of soundly rejected. Not only did we find that virgin females spent far more time associating with dishes overall, we saw that males, and specifically virgin male dishes, got far more attention than any of the other dishes, and we saw no differences at all between the behavior of females towards the different chambers between the focal treatments. So virgin females spent the same proportion of the time visiting each one of these 5 different chambers as they would as if they were mated females. So we were 0 for 3 in our best guesses about trying to figure out what's happening in the mind of fruit flies, which is always kind of exciting. And when we looked at the eggs, we saw that amongst the mated females, that they laid far more eggs on those that had came from male chambers, and against the virgin male chambers and mated male chambers having amongst the highest. So we have clear evidence from here that females are using cues, social cues, but it isn't necessarily copying. That what we suspect might be happening is that females are not are looking for male cues, and why they're doing that is something that we're going to be exploring in the future once we get out of lockdown. It could potentially be that males are better spending time finding in the wild, finding more time associating and finding better quality food. And so instead, females are actually looking where the males are, so they are offloading the hard work of finding suitable sites to males, or it could just be that they're attracted to cues for males for mating or remating purposes. So lots of sort of exciting avenues for this going forward. Our sort of second experiment that I'm talk about is some work that came out of Lucas Coede's master's work. And Lucas was really interested in sort of the social aspect of fruit flies. Fruit flies have become a really important model to sort of study social behavior. And Lucas came to me and sort of said, the thing I'm really excited about is social behavior. Oh, excellent. We looked at your mating interactions, things like that. He said, no, none of that. I'm interested in larvae. And larvae is something which, I've always looked at, but never have really seen. And he was strongly influenced by this exciting work by Mark Dombrowski et al. That was looking at group clustering that it happens in Drosophila larvae. And I'm sure everyone here that has cultured fruit flies has at some point probably seen this sort of behavior where these third instar larvae work together and sort of dig these sort of these wells. So collective behavior. I'll throw this video up in time. And this is a really interesting behavior because fruit fly larvae are for the large part sort of stuck in where their mothers lay them, and they may experience large amounts of competition at that site. And with more individuals in a particular space and time, there is more competition and typically lower survivorship. And this could, potentially lead to things that you might expect greater antagonism between individuals. But instead, we see that there is a certain degree of cooperation that happens, both in wild fruit as well as in our bodies. And this sort of happens at the third instar stage, where initially we have our larvae, which have done a really good job in our vials of depleting most of the good resources close to the surface and making that into a very soft and nutrient-rich waste product, nutrient-poor, waste product-rich environment. But lower down in the vial, beyond the range of any single individual fruit fly larvae, is untouched resources, things that they could access. And so we see fruit flies sort of accumulating together on the surface and coordinating their behavior to access this untouched, pristine resources below. Now, this is a really interesting behavior, but it isn't necessarily as widespread. That even when you look at vials, the vast majority of fruit flies are not engaging in this sort of behavior. So why don't they do it? Well, there's a couple reasons that have been sort of hypothesized. It could be because that in wild, that initial coordination of larvae means that they essentially have to stay still, and that might make them more likely to be predated or cannibalized. In lab, that can also sort of happen with cannibalization. That initial sort of group of larvae need to work together, and it takes a bit of time and effort to actually access that lower resource. So the rewards are not immediate. So it does take a certain amount of time for them and a certain critical mass to sort of create a deep enough well to access the food. And if some members of that group defect, well, your well may collapse. You've wasted a lot of energy, and you might even suffocate under all that soft, gooey medium. And so Lucas said, well, this may be a case where we could look at some factors that might make you more or less likely to want to cooperate with others. So the thing that he was particularly interested in was about potentially the use of kin selection theory, the idea that this working together, this altruistic behavior, is more likely to be seen amongst relatives as opposed to more distantly related individuals. That not only with kin or more closely related individuals being able to get direct benefits of working together, of access to food, but they get indirect benefits by the success of their relatives. So in this experiment, we sort of started off by creating a whole number of different families. Here's Lucas and my son at work labeling vials, where we created single male, single female families and collected all the sons and daughters as virgins as they were closed. And then we used these to create 3 different kinds of experimental treatments. One in which, in these treatments, we would use these to generate the eggs, which we would then place into vials and measure the behavior of the larvae. So in our high release treatment, we put in 5 males and 5 females, each of which came from the same family. So the coefficient of relatedness of every individual to each other would range between 0.5 to 0.25, with our estimated mean coefficient of relatedness about 0.3. In the medium relatedness treatment, we put in 5 brothers and 6 unrelated females, and in the low one, it's 5 unrelated males, 5 unrelated females. So we have a sort of gradient of relatedness between individuals in our treatments. We then counted out 200 eggs from each one of these individual combinations and seeded that into a new vial. And then over the course of 3 days, each one of these vials was observed in 3 different sessions. And in each session, the number of clusters that was observed in each vial, the number of larvae in each cluster, and the width and depth of each cluster was recorded. What we found was, across our 3 different treatments, was that generally speaking, when the coefficient of relatedness was higher, we tended to see more clusters being observed. We saw those clusters were larger in size, and they contained a greater number of larvae. So these are all sort of suggestive of the fact that individuals that are more closely related are better able to work together, or more likely to work together in this sort of cooperative venture. We then sort of follow this up with a sort of secondary experiment, or 2 secondary experiments, in which we placed in 50 eggs that were from our wild-type population, which were either red-eyed products that were either from the same kind of high, medium, or low relatedness kind of treatments, and we embedded them or combined them with a wide range, 150 eggs from one of our sister populations, IVBWs, that have ground pines. And so we wanted to see how this sort of group of 50 individuals that would do when they were mixed in a more realistic environment, where there's many different types of individuals. So in the first experiment, we measured the survivorship of our focal 50 eggs, and measured the weight of the males and females that had closed from each one of these vials. And in the third experiment, we did the same kind of experiment, and then we sort of went in on sort of day 3 and scooped out one using a sort of spatula, one cluster, and put that into a new vial and let them grow up, and so we could see how many individuals were in that cluster and how many of them were our focal flies versus our brown-eyed competitors. And we saw that there was a pattern where there was a higher degree of survivorship amongst individuals when they were the focus flies, when they were more closely related to each other. We saw no real sort of difference in terms of adult male body size, but we definitely saw that females that it closed on those from the high relatedness treatment files were larger, presumably because by working together they had access to more resources and thus were able to close greater. So there is evidence that that clustering is definitely adaptive, especially to those that sort of participate. And in our experiment 3, we found again another significant pattern where we saw that while there was no difference in the total number of larvae in the clusters we removed, that there was a greater representation of our focal wild-type individuals when they were more related to each other than there was a lower average relatedness treatment. So again, sort of suggesting that we find relatives working together more frequently. And this could be because of a couple of different reasons. It could be that kin are more likely to bear the costs of working together, especially in the initial sort of founding of a cluster. And alternatively, or not mutually exclusive, is the fact that Dombrowski et al. Found that creating these things is not surprisingly trying to dig a hole out of liquid food does require a fair amount of coordination, and we cannot match the motor profiles of the contractions that the cluster falls apart. And so potentially individuals are able to recognize their kin and work together, and or relatives are more likely to have similar motor profiles because they have shared alleles from their ancestors. So again, something that we're interested in sort of pursuing more in the future. My third project that I'm going to tell you about today is work that sort of comes from the joint work of Matt Maloney and Karen Newman. That is interested in sort of the problem of sexual dimorphism. Sexual dimorphism, I'm sure everyone here knows, is an absolutely incredible phenomenon where you have differences in the phenotype of males and females. Really important for many different reasons. It may be associated with difference in ecological use or associated with sort of sexual selection. The underlying theory is that for whatever reason, there are initially monomorphic species where males and females have, on average, the same phenotypes. They are different optimal phenotypes, and that leads to selection, sex-specific selection, that would lead them to evolve towards these peaks. Now, the problem is that often the traits that might be under sexual dimorphism are under joint genetic control, but they share the same genetic contribution. So you would tend to see sons and daughters having similar phenotypes. This phenomenon is called the intersexual genetic correlation, and is often why you can often tell which side it belongs to the grooms and which side belongs to the bride. Now, intersexual genetic correlation complicates the evolution of sexual dimorphism because an allele that might be favored when expressed in one sexual genetic background is at a selective disadvantage when expressed in members of the opposite sex. And so, as such, this will constrain the evolution, preventing them potentially from becoming as dimorphic as they could be, and causing neither of the sexes to really reach their full fitness potential. Now, the work that Matt and Kara did was influenced by this really interesting paper done by Stewart and Rice from my old lab at UCSB that was interested in trying to see whether they could artificially evolve changes changes in the magnitude of sexual dimorphism. And this is done on the LHM population of fruit flies, where there is considerable variation in both male and female body size. But as you probably know, female bodies are, on average, larger than males. Bill Weiss developed this really cool technique that allowed the efficient sorting of fruit flies using sort of a column of sieves here, in which each sieve contains these amazing sort of electroformed holes that are 5% larger than the ones below. So this allows the efficient sorting of fruit flies, nested size, of course, that can be separated out according to essentially their girth, how far do they go before getting trapped into one of these increasingly small holes. Now, over the course of 10 years or 250 fruit fly populations, Andrew either selected males in the sorted populations and either selectively made only the largest males with the largest females, concordant selection, or in other populations, the experiment evolved for small body size, so the smallest males to small males. In his experimental treatment, his discordant population, he picked the smallest females with the largest males, so going against the sort of the normal direction of natural the dimorphism forward apart. And after 250 generations, he found extremely little change. That the populations, although they did have a little bit of reversion of sexual dimorphism, it was nowhere near the extent to which that the concordance selection lines showed they could potentially have evolved. So this does sort of strongly suggest that the intersexual genetic correlation may be an important constraint on the evolution of sexual dimorphism. However, there is a different school of thought about how one could use selection to manipulate the extent of sexual dimorphism. The Delph and colleagues did a really interesting study about 10 years ago where they used a sort of artificial correlational selection, where they said instead of selecting the individuals at the phenotypic extremes and mating those individuals, that we should look at the intersexual genetic correlation. If we wanted to change it, we want to look for those individuals that are not at the extremes of the major axis, but are at the extremes of the minor axis, that are furthest away from the expected, from the pattern of intersexual genetic correlation. And we selectively mated those individuals, well then we might have a rapid evolution of sexual dimorphism. And they did this in the white campion, Silena latifolia, which is a dioecious plant, and they looked at the calyx width. And in two of their this system, there is a strong positive genetic correlation between the two. And then they imposed this artificial correlational selection on them. And in two of the three populations, they found a dramatic change in terms of the extent of the genetic correlation. That, especially in the earliest generations, they had a dramatic change and maybe some reversion later on. So potentially saying that it is possible to change the degree of sexual dimorphism, but you need to pose different tests. But here we have two different viewpoints. We have Stewart and Rice that is selecting flies on girth, selecting at the individual level, selection for decreased sexual dimorphism. On the other hand, we have Delph and al., which is selecting plants, artificial correlation selection, and selecting for both increased and decreased sexual dimorphism at the same time. These are really different studies, and how do we know what is sort of the takeaway, the sort of generalizable features of these? Well, we need a study where we could combine the best of both worlds, and we were in the unique position of having the same kind of flies, having the same kind of sieves, to sort of to do that experiment. where we combined some of the most relevant elements of both and tried to apply the delphinol protocols to the Stewart and Wright's techniques and systems. So in this experimental evolution project, we initially did a survey of our OHM population. We also did this in our parallel OHM-BW population, and we looked for that intersectional genetic correlation, and we picked the individuals that were furthest away from the major axis, furthest along on the minor axis. And from there, we pulled out 8 different families, which we mated in this particular array, generating us these particular combinations of families. You can see we had we mated individuals that had increased sexual dimorphism together, and the individuals that had less sexual dimorphism together. So we're selecting for both increased and decreased sexual dimorphism at the same time. In our control populations, we just randomly picked flies, but mated them in the same using the same sort of system of full family matings. We kept population sizes the same and treated them otherwise the same. So we did this for five in this initial generation to sort of generate both our control and our experimental populations for both our LHM and our LHM-BW population. And then each generation, we collected sons and daughters from each of those families, ran them all through the sieve columns, figured out which families had the most or the least dimorphic, and selectively mated them in our experimental treatments, or just randomly combined them together. And we did that for five generations. And then at the end of that, we decided to do sort of a fitness assay of the fitness of males and females for each of the families in the 6 generations. Now, in our initial survey of the population, so in these graphs, you can see several of them. We have the mean size of females in individual families is plotted here on the x-axis, the average size of the males based on essentially their girth on the y-axis. And we used ASRML, as in delphinol, with the pedigrees to calculate the heritabilities and the intersectional genetic correlations of them. And in both our LHMs and our LHM-BW populations, we found that was evidence of a positive genetic correlation. And we then sort of picked our control ones for our control populations, we picked the ones for our experimental populations, and we came back a couple weeks later and saw what happened. And we were absolutely shocked to see that after one generation of selection, that we dramatically changed the calculated estimate of intersexual genetic correlation. So we can see in our experimental population, we actually got a negative estimate of intersexual genetic correlation. In our controls, it still stayed positive. And in our we saw the similar pattern happening in our LHM-BW pair population. So in both these populations, one generation of this artificial correlation selection could fundamentally change the expression of the intersexual genetic correlation. Now, this is the point where in retrospect, you say, well, why didn't you stop there? Well, we wanted to see, if things could keep going. And so we did, and this is where things get complicated. Because although we had this interesting pattern, we still picked individuals that showed, in our experimental populations, that showed the most and the least extent of sexual dimorphism. In all our subsequent 5 generations, we saw a reversion to a positive intersexual genetic correlation. And that happened in both our experimental LHM populations as well as our LHM-BW populations. So really surprising that we sort of saw this change. And there's a bunch of reasons why it could potentially be. It could be that but through our strong selection, only picking 8 families in every generation, that we may have sort of depleted most of the genetic variation present in the population, and thus anything that remains is the patterns are soon going to be driven more by microenvironmental variation and or maternal effects. It could be the complicating effect of epistatus. Body size and fruit size is controlled by over 1,000 different genes, and from some work looking at hybrid vigor, that initially when you say cross 2 inbred lines, you can often get a very consistent phenotype, but when you cross members of that F1 generation, you get lots of different unpredictable phenotypes, and I think that might possibly be what's happened here. Or it could be the effect of that artificial correlation selection, where we, by selecting for both increased and decreased sexual dimorphism, that might be sort of complicated matters. Surprisingly, that when we look at the patterns, that whether both your parents were positive families, does not necessarily predict whether your offspring will also be positive. So we saw lots of sort of potentially interesting things to follow. It was an interesting study done in '72 by Bird and Schaefer that used sort of a family level sexual dimorphism thing on body size and on wing size, but only selected in one direction, and they found a very rapid change in sexual dimorphism in both directions. So this is also something we might explore more in the future, comparing simultaneous selection versus directed selection. Now, we also sort of remember at the end of these sort of 5 generations, we also curious to see, well, what are these populations like? What has happened, if anything, to the fitness of the males and females in these populations when we experience different kinds of selection? And so we conducted assays of male fitness and female fitness in all of our 4 experimental populations. We grew them up under standardized densities. We collected these adults, and we placed them into our standardized competitive environments, and measured daughter fitness as offspring production, and son fitness as a proportion of offspring sired in a competitive environment. Now, when we look at the daughters in these populations, we saw no differences at all between our experimental and our control populations. But in both our LHM-derived and LHM-BW-derived populations, we saw our experimental populations had a significantly higher reproductive success than our control populations, despite the fact that everything apart from the type of selection that we imposed, the choosing of parents, had been kept the same. So we do sort of see some potential evidence that our artificial correlation selection allowed males to get a bit closer towards their fitness optimum. So in the end, and this is the picture of Tara and Mac finally finishing this 7-month experimental evolution project, we find that there is common ground both between the work of Delft as well as between Stewart and Weiss, that we see that there is the potential for intersexual genetic correlations to evolve rapidly, but it does require specific kinds of treatments, and the plausibility of this kind of selection in nature is something we can talk about later. And we also did show that we could find some improvements or a better performance associated with this artificial correlation selection. So perhaps we were able to decouple male and female fitness between these populations. So the changes that we were doing on body girth may have had some indirect effect, which influenced their success. And so that wraps up this talk about that has been really sort of fascinating for me to sort of try out all these new different topics. And I want to thank Heather, Lucas, Tara, and Matt for sort of, bringing these ideas to me and working with me on sort of bringing them to fruition. And I thank you all for your time and attention, and I'd be happy to answer any questions that you have.
Well, thank you so much, Tristan, for a really, really fascinating talk with some very interesting, unexpected results popping up there as well. So, yeah, it'll be great to hear everyone's thoughts on that. And if you have any questions for Tristan, please do put them in the Slack channel now. So we already have a couple of questions from the audience. So the first one is, were you surprised by the estimates of RMF at the start of the experiment? And this person says, if they remember correctly, other estimates of RMF for body size in fruit flies are typically greater than 0.9.
We were somewhat surprised. That we were somewhat surprised. There is, of course, the elements about how much opportunity for a sort of mating is actually possible in things like the OHM line. So the underlying genetic variation that's present in the population is not something that we were able to control because we wanted to keep that. But that is actually a really interesting, because it was surprisingly smaller than I was kind of expecting. That being said, there are some other sort of issues when we set these things up for logistical purposes, because we were setting up all these families. We just put in males and females together, and we did not control the larval density, which could potentially have caused some blurriness in that pattern. That blurriness can also have led to some of the unusual results going forward. But yes, so doing this with wild-caught populations or with ones would be an interesting sort of complement to this, to see is it just the OHM that has this pattern, or something else about how we did this experiment.
Thank you. So the same person, I think, asks, if they recall correctly, Stewart and Rice argued in their paper that the Delft study applies a selection regime that is unrealistic and not how selection would work in nature. What is your view?
That's a really interesting question, and a very important element. So, to some extent, I think that they are absolutely right. That choices often happen at the individual level. But that being said, then in nature, you could also imagine scenarios where you could imagine individuals that are brothers and sisters mating assortedly. That's a fairly common phenomenon happening. Or if you have a heterogeneous environment, you could potentially have, under some circumstances, small populations that might be made of individuals that by chance happen to be further along. So those populations might potentially have the opportunity to diverge through in a parapatric kind of methods. But again, that is a really interesting sort of question, and actually we're playing around a bit with some of these questions about decision making under different scenarios. But yeah, I entirely agree that at least in fruit flies, probably that individual level and family level is in vials family level is not as easy to happen. But in nature, I think there's some plausible scenarios to see happen.
Sure, thank you. In your experiment on relatedness coefficients, do we anticipate lower dispersal morphs in higher relatedness clusters, or the opposite is expected as lots of animals actively avoid being inbred?
Well, in our vials, there really isn't much opportunity to escape much of anything. So green screen, so you can't actually see my green food. That you might there potentially could be differences in terms of that, and there is some interesting sort of work that's being done at other labs that is investigating this sort of phenomenon, to sort of look at that dispersal of things and how it manifests in terms of the things. But I would definitely think, that if you had sitters that were more related, you definitely would get more likely of clustering than sitters that were unrelated, and then rovers unrelated would probably fit at the least likely to work together, and rovers related would sort of be of that third or fourth combination. But yeah, I would say that would be an interesting element to look at.
Sure. I also had a question about the larval relatedness study. I think you showed some figures where the degree of larval relatedness had different effects on the dry body weight of females emerging from that larvae compared to males, and I wondered if you had any idea why it would be sex specific like that.
That's a good. There was a trend. There was definitely a sort of a trend that our higher, the intermediate one was not significantly different than high. High was not significantly different than control. It could just be like the precision of our measurements and that small variations in male size might not be sort of picked up well in this particular assay, while females have a greater capacity to respond to favorable conditions. But generally speaking, female size is often more strongly influenced by nutritional quality of the developmental environment than males. So these males could be doing other things. And it'd be interesting to look to see the fitness of these target larvae that grew up under these different scenarios. So you can actually see the real fitness impacts of this. So we see this higher survivorship. We see larger females, which are potentially more fecund. But it'd be also interesting to see, are those males also sort of benefiting compared to the other ones?
Sure. So we have a question here. Are you confident that the larval cooperation is truly cooperation?
Yeah. As much, in as such that they're working together, and all the limitations of our language and our understanding of what they do. They are working together. Now, are they working together better because they are more similar phenotypically, or are they working together because they can recognize each other as kin? Our results are still ambiguous on the subject. Other work that Lucas has done has shown that larvae do seem capable of detecting chemical cues that can recognize whether an individual is a relative or not. So we have some evidence, and there's evidence that larvae and definitely fruit flies can detect and recognize kin versus non-kin. But yeah, this is a sort of a jumping off point for looking at what's happening. So there are lots of different sort of ideas that we've sort of been floating around for future projects when we get out of lockdown that could look to help distinguish between sort of these competing hypotheses. But we were just really interested to see this is a really interesting pattern, and one of the great things about the fly community is that you see that we take inspiration from each other's work and build upon each other. So I would love to talk to anyone or hear from anyone that sort of has thoughts about this.
Thank you. So maybe one last question, as I think we've gone slightly over the hour. But do you expect to see the well structures formed, the depressions formed by the larvae in real fruits, or as well as you see in the lab vials?
Yeah, definitely in vials, you get it is easier to see them. We only counted they only measured the ones around the side of the vial where there's that sort of long structure. But you do see these in real fruits. They do sort of work together to soften it up, because even a fruit that is rotting initially needs a certain amount of fruit flies to sort of work it, to sort of make it palatable and make it digestible. And while they're doing that, they're excreting all their wastes in there. They're making it into a sort of toxic environment, and the only good environment that's left might be sort of down below. So this is a phenomenon that I've left a bunch of rotting fruit in my backyard, and it is something you see happening both in the lab. So it is a real phenomenon. Whether it's magnified in our environment, probably, but I think it still has some interesting elements that hopefully capture some imagination for those out there.
Sure. Thank you so much. Yeah, and thank you for just such a really fascinating talk, and really shows the power of the humble fruit fly in answering these really important evolutionary questions. So thank you so much. And yeah, so now we have a few weeks break. We hope to see everyone again on the 28th of July, where we will be joined by Dr. Yasser Ahmed from Syracuse University. So please follow us on Twitter at EcoSeminars, and keep an eye on our Slack channel for more details about upcoming talks. Thank you all for tuning in, and goodbye.